Welcome to Food for Thought, a podcast from the nonprofit NutriSafe, where we talk about the connection between food, health, and chronic diseases and what we can do to create healthier lives and communities. I'm Simran Rogers, your host for today, and I am an upcoming senior at Northville High School and have a huge interest in learning about chronic diseases and diabetes. So today, our conversation will be about diabetes, and we're just going to be discussing that important chronic illness in modern medicine. When most people hear the word diabetes, they think about blood sugar, insulin shots, or maybe just avoiding sugar in general. But diabetes is so much more than that. Scientists now understand diabetes as a disease that affects nearly every organ system in the body. It changes how cells communicate, how energy is produced, how blood cells function, and even how the brain processes information. More than 500 million people worldwide are living with diabetes, and that number continues to rise annually. It is one of the leading causes of kidney failure, blindness, heart disease, lower limb amputations, and strokes. Today, we're going to be going all the way from the pancreas to the microscopic level of cells and molecules to understand exactly what happens inside the body when diabetes develops. Let's start with energy and the body's energy economy. Every second, trillions of cells throughout your body need energy to survive. Your brain needs energy to think, your muscles need energy to move, and your heart needs energy to beat approximately 100,000 times every day. Most of that energy comes from glucose, which is a simple sugar produced when carbohydrates are broken down during digestion. After eating a meal, glucose enters your bloodstream, but there's a problem with this. Glucose cannot simply walk into most cells. It needs help, and that comes from insulin. For those who don't know, insulin is a hormone produced by specialized beta cells located in clusters called the islets of Langerhans inside the pancreas. The pancreas sits behind the stomach and serves both digestive and hormonal functions. As blood sugar rises, beta cells detect the increase. They release insulin into circulation, and that insulin travels through the bloodstream and acts like a molecular messenger. It basically just tells the cells, open your doors, glucose is arriving in the cell. So now let's talk about when insulin at like the cellular level once it's in the cell. So if we zoom in to a certain cell, the cell membrane contains insulin receptors. Think of these receptors as locks, and insulin is like the key, and how insulin has to go into the receptors for it to open. When insulin binds to the receptor, an incredibly complex chain reaction begins, basically like the key in the lock. Scientists call this a signal transduction pathway. First, insulin activates receptor tyrosine kinase. Then, proteins called IRS molecules become activated. Then comes the P13K signaling. Then AKT signaling. Finally, glucose transporters known as GLUT4 moves to the cell surface. These GLUT4 proteins act like doors that allow glucose to enter. Once glucose enters the cell, it can be used to produce ATP, the universal energy currency of life. This process occurs billions of times every second, and most people never think about it. Now, let's talk about what happens when the system breaks. So, there's two different types of diabetes, type 1 diabetes and type 2. Right now, we're going to be talking about type 1. So, this type of diabetes is an autoimmune disease. Normally, the immune system attacks dangerous invaders like bacteria and viruses. But in type 1 diabetes, the immune system makes a catastrophic mistake. Immune cells begin attacking the body's own beta cells, and scientists still to this day do not know the exact cause. Genetics play a role, and environmental triggers may contribute to getting type 1 diabetes, and certain viral infections are still being investigated, but regardless of what exactly the cause that's being researched or what exactly it is, the effect and the result is still the same. The immune system destroys insulin-producing cells. Eventually, insulin production falls to dangerously low levels. Without insulin, glucose cannot enter cells efficiently. Blood sugar rises dramatically, and meanwhile, cells are literally starving because they are not getting the insulin that they need. Imagine standing in a room full of food and not being able to eat any of it. That is essentially what happens to cells during uncontrolled type 1 diabetes. And uncontrolled meaning without any medication or any help. So one of the most dangerous consequences of untreated type 1 diabetes is diabetic ketoacidosis, often called DKA. Since cells cannot access glucose, the body switches to emergency fuel sources. Fat stores begin breaking down rapidly. The liver converts fatty acids into molecules called ketones. A small amount of ketone production is normal, but an excessive amount can become dangerous since ketones are acidic. As ketones accumulate, blood becomes increasingly acidic. The body's pH levels begin to fall, and just before we continue, just remember that the human blood is normally maintained with an extremely narrow range around pH level of 7.4. So even small changes to that 7.4, give or take a little bit, can be dangerous. As ketoacidosis worsens, symptoms can include being extremely thirsty, vomiting, rapid breathing, confusion, dehydration, and even worse, a coma. Without treatment, DKA can be almost fatal. So that was type 1 diabetes, the diabetes that people are normally born with. But now let's talk about type 2. Unlike type 1, people with type 2 diabetes usually produce insulin. The problem is that cells stop responding properly. This condition is called insulin resistance. Scientists compare it to repeatedly hearing the same alarm until you eventually ignore it. At first, cells respond normally, then responsiveness declines. Then the pancreas notices rising blood sugar and compensates by producing more insulin, and even more insulin. Some people with early type 2 diabetes actually have insulin levels several times higher than normal. For years, the pancreas works overtime. Eventually, beta cells become exhausted and the insulin production begins declining. So it starts by producing so much, but eventually it's not doing a lot and it just ends up stops to work. And stops working and insulin levels begin to decrease and decline. But then blood sugar levels will rise further from this, and then at this point, full diabetes can develop. So I'm sure all of you know what obesity is. And so what we're going to be talking about now is the role of obesity and what it means. So for decades, scientists viewed body fat as simply stored energy. We now know that's completely wrong. Fat tissue is actually biologically active. It functions like an endocrine organ. Fat cells release hormones and inflammatory chemicals. But these chemicals include tumor necrosis factor alpha, interleukin 6, leptin, and resistin. When excess fat accumulates, chronic inflammation develops. Inflammation interferes with insulin signaling. The insulin receptor becomes less effective and glucose uptake declines. Insulin resistance worsens, and researchers now consider that chronic inflammation one of the major drives of type 2 diabetes. So now we arrive at one of our most fascinating aspects of diabetes. High blood sugar doesn't just sit harmlessly in the bloodstream. Glucose molecules begin attaching themselves to proteins. This process is called glycation. The resulting compounds are called advanced glycation end products, or AGEs. AGEs. Scientists often describe AGEs as molecular trust. So ACE's damage is blood vessels, nerves, kidneys, eyes, skin, connective tissues. They increase inflammation, oxidative stress, and they accelerate aging. The longer blood sugar remains elevated, the more ages accumulate. That is why long-term diabetes becomes so destructive. So the leading cause of death in diabetes isn't necessarily the blood sugar of itself. It's just a causing factor. It's cardiovascular disease that actually does it. People with diabetes have dramatically increased risks of heart attack, stroke, coronary artery disease, and peripheral artery disease. High glucose damages endothelial cells. These cells line every blood vessel. Healthy endothelial cells help blood flow smoothly. Damaged endothelial cells become inflamed, and cholesterol begins accumulating. Plaques form, blood vessels narrow, and eventually blood flow becomes restricted. If a plaque ruptures, a clot may form and completely block circulation, and that blockage can kill heart muscle or brain tissue within minutes. So now that we've talked about the cardiovascular part of it, let's talk about kidney disease and more specific, diabetic kidney disease. So your kidneys contain approximately 1 million filtering units called nephrons. Every day, these microscopic structures filter enormous amounts of fluid. High blood sugar forces kidneys to work harder. At first, they compensate. Over time, damage accumulates. The filtration membrane thickens. Scar tissues form, and protein begins leaking into urine. Doctors often measure albumin levels because albumin leakage is one of the earliest warning signs. If damage continues, kidney failure may develop, and many dialysis patients often develop kidney disease because of diabetes. So perhaps the most surprising discovery in recent decades is the relationship between diabetes and the brain. Scientists once believed insulin mattered mainly for blood sugar regulation. We now know insulin also influences learning, memory, neuron survival, and synapse formation. The brain contains insulin receptors. When insulin signaling becomes impaired, cognitive function may decline. Students consistently show increased risk of dementia and Alzheimer's disease among people with type 2 diabetes. Some researchers have even referred to Alzheimer's disease as type 3 diabetes. While not an official diagnosis, the phrase highlights how strongly metabolism influences brain health. So, fortunately for this, treatments have advanced tremendously when it comes to diabetes. Continuous glucose monitors can track blood sugar every few minutes. Insulin pumps can automatically deliver insulin for the cells that are not able to receive them. Artificial pancreas system combines both technologies. Meanwhile, medications called GLP-1 receptor agonists have transformed diabetes care. So these certain drugs and medication and these treatments can help lower the blood sugar, improve insulin sensitivity, slow digestion, and reduce appetite. Researchers are also exploring stem cell therapies, gene editing, and beta cell replacement strategies. For the first time in history, scientists are discussing the possibility of functional cures. So, before we can fully understand diabetes, we need to understand what normally happens inside the body after a meal. Imagine you've just eaten a bowl of pasta, a sandwich, or a piece of fruit. Within minutes, digestive enzymes begin breaking carbohydrates into smaller sugars. Eventually, most of those carbohydrates become glucose. Glucose enters the bloodstream and causes blood sugar levels to rise. This rise in glucose is detected by beta cells in the pancreas. But here's where things become really interesting and fascinating to many people and scientists. Beta cells are essentially biological glucose sensors. When glucose enters the cell, it undergoes glycolysis and cellular respiration. ATP production increases, and the increase in ATP causes potassium channels to close. This changes the electrical change across the cell membrane. the calcium channels open and they flood into the cell. Calcium floods into the cell. And this sudden increase in calcium triggers insulin resistance. Think about how incredible that is. The body has evolved an electrical monitoring system that can detect microscopic changes in nutrient levels and respond within minutes. Once insulin is gone, glucose begins entering the cell. Inside the cell, glucose enters a pathway called glycolysis, which literally just means sugar splitting. One molecule of glucose broken into 2 molecules of pyruvate. This process produces ATP and electron carriers called NADH. The pyruvate then enters the mitochondria and eventually enters the Krebs cycle. And this Krebs cycle generates additional electron carriers. These electrons travel through the electron transport chain. Finally, oxygen accepts the electrons and ATP is finally being produced. One glucose molecule can ultimately generate approximately 30 to 32 molecules of ATP. This ATP powers virtually every activity inside your body. Every heartbeat, muscle contraction, nerve impulse, and even every thought. Without glucose metabolism, life would stop within minutes. So one of the most important tools doctors use to diagnose and monitor diabetes is called hemoglobin A1C, often abbreviated as HbA1c. Most people know it as A1C. To understand it, we need to talk about red blood cells. So, red blood cells live for approximately 120 days. Inside each red blood cell is hemoglobin, and this carries oxygen throughout the body. When glucose circulates in the bloodstream, some of it sticks to hemoglobin. The more glucose present, the more glucose becomes attached. Scientists measure the percentage of hemoglobin molecules coated with glucose. That percentage becomes the A1C value. Think of A1C as a 3-month report card for blood sugar. Normal A1C is below 5.7%. Pre-diabetes range from approximately 5.7% to 6.4%. An A1C of 6.5% or higher is one of the criteria used to diagnose diabetes. Unlike a single blood sugar measurement, A1C reveals long-term glucose exposure. Doctors love this measurement because it tells a story. It shows how well glucose has been controlled over weeks and months rather than minutes. So one of the biggest reasons diabetes becomes so dangerous is oxidative stress. To understand oxidative stress, imagine a factory producing energy. Every factory creates waste products. Cells are no different. During energy production, mitochondria generate molecules called reactive oxygen species. These molecules are highly reactive. They can damage proteins, DNA, and cell membranes. Normally, antioxidant systems keep these molecules under control, but diabetes overwhelms these defenses. High glucose causes excessive production of reactive oxygen species. This results in widespread cellular damage. Scientists often describe oxidative stress as a molecular storm occurring inside cells. So now lastly, we're going to be talking about the genetics of diabetes. One question researchers have asked for decades is simple. Why do some people develop diabetes while others don't? Part of the answer lies in genetics. Type 1 diabetes is strongly associated with specific immune-related genes within the human ligand antigen or HLA complex. These genes help regulate immune function. Certain HLA variations increase the likelihood that the immune system will mistakenly attack beta cells. Type 2 diabetes involves hundreds of genetic factors, and scientists have identified genes associated with insulin production, insulin sensitivity, fat storage, appetite regulation, inflammation. But however, genetics are not destiny. Many individuals with high genetic risk never develop diabetes. Environmental factors also remain critically important. So, diabetes is not simply a disease of sugar. It's a disease of metabolism, a disease of inflammation, of cellular communication, and a disease that can affect nearly every single organ in our bodies. Yet, it is also one of the most intensely studied diseases in medicine. Every year, researchers learn more about insulin, genetics, inflammation, and the body of chronic illnesses. The story of diabetes is really the story of how interconnected the human body is. A small hormone produced in the pancreas can influence the heart, kidneys, eyes, nerves, blood vessels, and even the brain. Thank you for joining us for this deep dive into diabetes and chronic illnesses. Stay tuned for next time, as we will be having an interview from a doctor that actually did have diabetes. Until next time, stay curious and keep exploring the science behind health. This is Simran Rogers from the nonprofit NutriSafe, ending this second episode of Food for Thought.